I don't think it's
All right, now it's running. You wanna speak the intro again?
I guess.
Speaker true.
This is Genetics Lab on October 16th on Thursday.
Perfect.
We'll just put under. Yeah. Probably Crow.
Do you want to do
Probably Crow. Even though she's taking forever.
I have no part. Like you said, we're missing.
The second positive control. Also, you might want to, just FYI, you might want to cut that in half, because otherwise, if the paraffin is too big, it will kind of bulge up. So you might want to cut that in half.
Okay.
Yeah, just fill it to where it connects on both sides. Apparently, it doesn't matter how much you do. Coordinate our G's, I know whatever you do it.
You guys have the other positive?
Thank you.
We have one.
It's like a strip of red.
I took one of them. I was.
I would do more. Wait, yeah, I would do a little bit.
Do you have the positive strip of positive controls?
I put it back. Do more on the other side.
You did this?
I guess it's only two that I can make up. You can
I don't really think it matters how much you do, even though I think that's but he says it doesn't. Hurry up, tack on. No, it's for volleys. Run the rig.
My recommendation is your position is so that you will need it later.
How can we fill this too?
It should.
Position it already so as if you were about to load it. Because right now you're just running as black, right? Just running that.
If you're running the test,
Like for example, the positive is the red or the black one?
I'm gonna say red.
Red.
Okay, then you want to then turn it around upside vice versa, because it pushes away from the negative toward the positive.
So wait, negative.
No.
Where do you need to go on the bottom?
Boy.
So,
Look for the top modella. You're good.
I can hear it, but I don't see the numbers. Oh, we're going. I see
them. Wow.
Okay, you get an electric current here. That's good. What about you?
Are you just outside?
Did you
How does
test
it
electric current? Did it work?
Yes.
Like
Just
look.
out of body, right?
Well, yeah, look at the black part, and you should see like the bubbles going up.
Yeah. I thought I was going to do like a jacuzzi type of thing.
Hot tub? No.
Yeah.
I don't really know how to turn this off. Oh, okay, you just hit run again. Okay, you should be good to take it off.
Do you have
You do it first, so I don't get that.
Using logic, use the same one as
Well, mine's going to want to wash it past the point
I don't think we need the formula. If it comes as a 6x ball, right? You know, on the tube, the loading value.
Gel goes in. Yeah. Yeah.
Give it size. Gel loading die, 6x. So, in the actual sample that you loaded, it has to go to 1x. If this is a 6x one to begin with,
What do you guys do then?
you would determine how many of that with how many of your sample.
And our total volume would be 20, or was it?
Not necessarily, like the volume that you pipe it in is your choice.
You really have to make sure the
Right?
volume is okay.
But
But don't we need to know that?
I'm scared of it.
I'm scared too. I don't know if you get
I don't either.
one microliter from here,
Yeah.
5 microliter of your sample together would mean that this loading die is
What?
because it comes in 6x,
Right.
right?
Sorry, you just
So,
scared me.
are you guys okay?
Yeah.
I don't know.
Okay,
So you didn't rip?
Check it out. So,
Wow, okay.
you can
Apparently my
either
imagination,
do
like, actually,
1
like-
microliter little bubbles
saw
here
it.
and then mix
I thought I
5
saw it.
microliter.
I thought it was you.
I didn't feel anything.
I really thought I saw it right there. I had to look away.
But I would
I
say
didn't,
one.
I didn't feel it.
No, you're fine. I thought I saw it, and I was like, oh, I can't watch.
Again, like, The library code, I write this every
I'm
day.
sorry.
Maybe it's you come back to
Yeah,
explain
it's just,
yourself.
this is supposed to be a guide. You cure starting next week, you have a single number. I'll just tell you, please, running on the tell like.
Oh, I ain't touching
And
that.
then you figure out the answer. So this is why, just
It did rip.
6x
What's up, Bob?
Yeah.
logically tells you, okay, one piece, like one part, we have 5 parts of the sample.
So I did
would
see
be
it
a
rip. I thought-
X
Ours didn't even
in the actual
feel it.
solution.
It's okay. I thought I saw the ball rip.
She's like, hello.
It's okay.
Oh yeah, sure, okay, then.
We just won't use that
Did
side.
you put those in smaller ones? Oh, awesome, thank you.
We just won't use it. I'm just gonna put this
You think it's good?
What happened?
Nothing.
Nothing.
You see, if you keep it swimming it like that, it will over time shift the drift.
Yeah, we got
I've seen how. Yeah, because otherwise it will shift the drift.
Oh!
Okay. Do you have a spatula?
Yeah.
Can I just get a spatula? We get a spatula. That's
Stupid.
no, just pick it.
No. Lisa, you don't understand.
So what do we do with ours? Ours is in there too, but ours is full.
It's full.
Yeah, so ours is not doing that.
You did not scrub. You were looking at
Ours doesn't have the tape.
I hate the tape. It's my nemesis in this one.
Do we have to take ours out? Like, they're
I think so. Open muscle wobble.
Yes. No, Lisa, I'm not making another gel. I refuse.
You break it, Raymond?
No, I don't want to break it. That's why I'm not making it.
Oh my gosh.
I'll do it, but I swear, if it breaks, I'm not making another gel.
We're just gonna. Are you sure?
Yes.
Okay, here we go.
Lisa, I'm scared. Lisa, I can't do it.
Just hold it for a bit. I'm flat.
I can't do it. Lisa, you do it. And if you rip it, it's all your fault.
You're the grad student.
Yes.
Do we use the yellow tube or something else?
Did it work?
Well, loading that
One
is not the ladder.
Well, yes, like.
The other one's
Yeah, that's good.
Oh, that's the loading that.
We don't need that machine. Yeah, you don't
Loading
need
that is something you use for area of the samples.
It's so stressful for no reason.
Yes, ideally, always start the yellow. By the way,
I need to add more tea.
Yeah.
That is a DNA lab.
It has to come.
Barely
All
covered.
issues that is.
This is where you do it.
Yeah.
You'll see it.
Too much?
Dr. D. Using the actual.
Good point. Yes, I'll help you with that.
You too? It looks pretty wet. That's good enough. Wet enough. Okay.
That was new with the
Oh,
well.
natural.
The last time.
So arrange this.
Oh yeah.
Very much should have had like 12 units of
It does tell us
stuff
what
with
how they
my
were
first.
Okay.
And I was like, I don't think I
All right,
can.
good.
So you're gonna get some
You're right.
Okay.
That
So
would
do
work.
get your piece of caramel. Get your samples ready and do the loading time.
You could probably just put a small one and put a pipette tip, a fine pipette tip on the side as a holder, whatever you prefer.
No.
As long as the.
No, no,
Okay, just try to.
ladder.
And we'll try it out. One pipette tip on the side. Take a look. Okay, should be a good holder, right? And did it break? Or
Wow.
were you able to?
What do you even care?
No, I do care.
One of the walls ripped, but only one of them. Only one of the walls ripped.
Like one of the, like a car.
No, just one. One singular one.
Okay, yeah, just avoid that.
Yeah.
Oh, this one is frozen. What tip do we use to put in the well?
Yeah.
Fresh whatever one you like. But
I like, last time I punctured it, you told me that
We're just using the first row.
And one. I mean, we need to do eight, so we're gonna probably go the first six, and then we're going this way, because I don't know if that route goes into the other one.
especially if they have like the super fine tip, but you know, you have all the time in the world. Just carefully put it down and you know, puncturing or the yellow one is just fine, man. The yellow one is just fine. If you do puncture one, well, I think you have at least one, maybe even two
Well,
replacement.
unless we're running two tops.
Sure. It's very nicely sealed here. Took a good part. Okay, let's see.
We're not using the top get control.
No, put
Okay.
Let's try it.
Okay.
So,
Did you get the ladder?
Maybe we'll take a yellow one. This might be different. Hold on, I'll try to get a yellow one.
I'll have you do it. Okay. Yeah.
Why did you want to?
So put it in the one on the left.
Yeah.
This is
Put
not
the two.
trash. I put in my phone.
Yeah, we'll put one over the other side as well. Can you get us another yellow one? And then it's. But again, don't go beyond 60 volts, because this one machine really only does 60 volts. Otherwise, you'll melt the gel. Oh, look at that. Perfect. Yes. So you can just put the gel here, and then.
Thank you.
Yeah. This is water.
Some of it is infected.
Oh. So
Yeah, so ladder.
May I make a little recommendation?
So 5 millimeters each. So we're not trying to do this.
Help yourself
No,
about
5 millimeters loading die. and
So
then
many
yeah.
people basically type it like this, and you see you don't have any stability.
Oh. Yeah, that's not my expensive. Not big, I should.
Careful.
So you have
Do you need a fresh?
Do 5 on the paper, and then 5 for loading die on the paper.
Okay. Just keep the dots reasonably apart, because otherwise you contaminate.
We have more area.
Exactly. You have plenty of paraffin. You can use that paraffin all the way. I just suggest if you kind of, because otherwise if it's too big it will make
What are all the letters?
bubbles.
So bees, bacteria.
Oh, you can.
Okay, I got it.
Suck it up with
Plant, animal, okay. Yeah.
Yeah, exactly.
Boy.
Oh yeah, we should have given these.
Oh my god.
Sounds good. Oh, this is gonna do some Christmas on
But
that.
that one was as dark.
Wait, did you use it all?
Huh? I know, we're gonna use that. We need new hair or something. We forgot to do
I was like, this looks weird, but.
I was like,
We were ready.
wait, why does it look like that? And I'm like, wait, no, that's right.
Do you pack with the little
Are you sure?
Do you guys have
So it doesn't really matter which one you do first.
No, just they say if they give you
You just
I saw that.
You just kind of mix it together. You do like a drop. You do like a drop of each, right? Yeah. You get more.
Well, she said we should have
Yeah,
that.
we're gonna basically wash.
I
Like,
think
So,
yeah. Then it's
That's what we say.
like if it's outside, it's I mean, like it's never happened before.
It's 5.
Okay.
Here's the loading guide, Ella.
Okay.
So here are the instructions. If you
You have to do all.
Sure does.
Yeah.
You don't have a
I mean,
Anyone else?
here you go.
I'm gonna keep this in left.
The normal one, so make sure it's normal.
They won't come on. You gonna do some live work afterwards? I'm just fine. Are you gonna do some live work?
Oh, wait.
Oh, she just,
I said put this at 5 and 5.
Okay, let's put it back on.
I don't even think spit it back out. Take it in there. So make some love.
Officially here. There's one.
Oh my gosh, we get the. Yeah, just let me do probably.
That's what I'm doing for you. I'm gonna do a large.
Okay, we're gonna do the animal one next.
Oh boy. Oh my god, this is really good. I don't know what it was. Dude, it's like you didn't have this eating.
I'll start with the regular size.
I just don't have petrol.
Oh, which is, yeah. If you want, one of them in blue. There's blue there too. What's the blue thing, bro? Sure. All right, how's your loading?
Good enough.
I've been trying to prepare for
Do you guys have a loading guide? Yeah.
I just don't see, other than the first one, I don't see it.
Well, I think it's almost because the dust is not as, like, it's not as dark as the ladder. Because this one was the ladder, so that's a lot darker.
Is that's a die?
Yeah.
Okay. And you use 1 and 5, right?
Correct. And the ladder was kind of purplish, so when we mixed it with the dye, it was like a lot darker. But
Sure.
then this stuff isn't as dark because
these our samples are like clear.
Yeah, of course. It's just, it should definitely be darker. There's something going on. No, I'm
sorry.
Did you dilute this, or did you just make it 1 to 1 or 2? No. We're just like this.
That's fine.
Would you like to do that? Just take this. Yeah, that's the original concentration. May I make a recommendation?
You know, I was just
I don't know what it is.
So this is the color of our sample and it's kind of light.
Yeah, it's like really light. It's like a light purple.
Yeah, but then even that should be visible there, so I'm just wondering if it immediately
But I'm
resolves
getting
for you, because I can't
It's
see
kind
any.
of, yeah, it just like, I don't know.
Can I do one of them just to see, because in the worst case, you can, if you know which
It
sample
might
is
in
which.
fact be me, but
I don't think it is because like see how light it is?
Yeah, which one would you film now?
This one would be in number 5.
So 1, 2, 3, 4, 5.
Well, you can see that one.
Yeah, but even that one is.
whatever you need. Maybe it's our buffer.
No,
I can't see it at full now,
I'm afraid I know what this is.
but
I think either from last week or during that waiting period now, I think the gel has, like the little wells have broken open at the bottom. Because this is exactly what it looks like if something seeps out exactly as much. Because, you know, like, you could see it initially, and now it's basically vanished. I'm afraid that's exactly
How much of our
No, we'll do. We'll actually do it.
Just
the
the
right
right.
test here. I'm gonna put it into the only pure loading die, 5 milliliters. By the way, you would need 6 of them, right? Because one but I'll just take it pure and put it into the die. Well, and if that also disperses, we know it's
Okay, I think if they have private donor that does
I just think it's hard for me to see like where it's in.
But that's a thing maybe.
well, you know what? Can you
Yeah.
can you treat this as
Yeah, that would be
Perfect.
let's treat it as a 3x buffer, so you will
P.
oh no, you actually triple.
Do we want to
Oh, yeah.
I can do one more and then we can do one.
You take 5 microliters of these again because you have plenty in each of them.
Yeah.
5 plus 15. No, my apologies. No. 5 plus. It would be 5 plus 2.5. That would be the correct ratio. Because then you should see better. Yes. Can you do 5 and 2.5? So you can take a fresh one of those. Make 2.5 microliter bubbles.
When you're done with the loading die.
I mean the.
Or better, let's make it 3 microliter. 3 microliter. Oh, no, actually,
This goes to 2.
this
So.
is only goes to 2.
Stealing someone
Take
else's.
3 microliter bubbles, and then do 5 plus 3 and a little bit.
Okay.
And
But
because
also,
you remember what the sequence was, right?
Correct,
Then
yes.
you just redo them with 3 microliters of the loading die, and then 5 microliters of. Yeah, let's do 3 to 5 because that's easier to find.
does it matter that since there's like already stuff in the wells, or just like be able to
My friend,
keep
yeah, I'm
loading
pretty sure
it
it is.
after
It is.
Okay.
Yeah, you just start from the beginning again in the same order. 3 of these.
Okay.
We have collected them.
Yeah.
And 5
sneak on this.
Okay.
Yeah, because otherwise you will not.
Yeah.
If you
Okay.
run this, it wouldn't be able to.
How many microliters did you put in this last well?
You
Five.
have 5 microliters.
Five.
I mean, we have spare.
Well, because that should be our extra positive sample, our last well.
Should we just run
Like the one who
positive
said-
and
Oh yeah, sure. Okay, then you know what? Then just add to it because all there is now is just some of that loading die. The loading die itself is inert, so just add on top of it.
Okay.
It won't.
You're done with the loading die.
affect the actual sample.
Okay, sounds good.
Thank you. So this is that.
All right, how do you guys doing?
We don't have much left in there.
And
There's the wrong one.
We need
And
more
have
loading
if you
die.
want to use more loading die, add
This
it.
might not be enough.
You also using two loading die.
I got literally every single drop.
If for 1 to 6, yes, but it's because some of your wells are also super big. Oh, I see. Okay, good. Yeah. But if you want, you can go a little heavier with the loading. Why don't you just add another microliter? Before you suck this up, just add 1 microliter of the loading die for 1 extra one for each of the samples. So do 2 5 from now. It's just
What?
for some reason our calculation is a little.
Was it like
Somebody-
that last year?
No, somebody must have diluted it.
Let's do it, Bill.
It's the only way I can imagine. But yeah, it's good that we saw it ahead of time.
Yeah. A little strand cut. After I push out the final part of the
I
strand, but
think
I'm
what's going on is you have to gel.
So bacteria.
Really?
You see?
what's happening?
You see?
What now?
I don't think so.
You see the wells are actually at the bottom. So
There's ours not Home Depot. It's the same way, so like Home Depot,
Okay.
it just makes me
This
gonna
is
say,
8.
Ours are sitting in the
So
well,
that's
so
the last.
presentation will fit out.
That's not black.
No, it's red.
So it's like they're like
You're not here, hold on.
unless I poke through all of them.
Wait.
What are you owing? You went, oh.
Okay, we're actually doing this. No, that's
Because if you had now gone in with a positive, you would have come from the other.
No, we can't.
Positive and negative.
Seriously?
No, we're good.
Oh yeah, that looks good.
Yeah, I have a loading die.
Okay. So, Starlight.
I have a loading die.
If you need more loading die, we have plenty here. Loading die is something that.
We've already taken like $300.
May I ask those that haven't started yet, why don't you add 1 microliter of extra loading die to each of your bubbles? Because it seems to, don't worry, if in your case one can see it nicely, so maybe you got the right number to begin with. But actually that looks pretty dark too. For some, 5 of that plus how much of the? Oh yeah, okay. That's because you see the loading die comes as a 6x. Normally you would use 1 microliter loading die and 5 microliter sample to get it to a 1x. But again, in your case, this for some reason is not concentrated enough anyways. We see that in the other gels. So while you're overdosing here on this loading die, it's not the worst thing to do because loading die is inert. So if you do too much, all right, then you do too much. There's nothing with loading die that can happen. So you did 5-5, fair enough. Then you're on the safe side to begin with. Need more? Loading die? Yeah.
It worked out. Wait, it worked out.
They did actually 5-5.
It's fine.
Beautiful. Which is a little
Do we
much,
just
but
put the ladder in? Ours might have got stacked. Do we put the ladder in just or like because when she put it in there, you first it might it has milliliters. So
that's exactly that.
so it might have just broken down.
Better gotten to do well.
So first of all, when you pipe this, since you have bubbles in there, this will make it quite difficult. You might want to put it back and suck it up to the bubbles. But because bubbles means that when the bubble comes, then they literally bubble out.
That's such a good well last time.
I wasn't sure enough.
Shall we make a fresh gel? Just this one is very quickly. The small one. Your choice. I mean, if you say it was only 1 or 2 of them that are broken,
We might need 5 again.
No,
so it is. But if it's like the majority, then
Oh, you want to
might be no.
Because we did one like gel
It's your call.
I say load that one and let's see what happens. But
Maybe this will work.
this would be 2. Okay.
Yeah, but
And then set it back up.
Oh, you're smart doing that.
I know. I've learned from my master mistakes of the bubble.
Here, I brought that 5 microliters.
alright, how you guys
Something is really dark. It's loading
doing? By the way, did you
It's sitting in the well here.
that
Okay. How do you want to do this one? Do you want to start with this before I
We're doing positives and negatives.
do you want to do positive?
It's all in
What's that? It's all like our powers don't for us. Yield anything.
Weird. No. I think quite frankly, it's good that all of you guys, yeah, so please make sure that everybody gets a little bit of pipetting activities under their fingers. Zachary,
You want to do it?
definitely do some pipetting too. Because for next week you'll need it for the cure. Because you're gonna be reshuffled into
I have
new
a loading
teams.
die. Nope. Oh. Oh.
More loading done?
Oh,
Sorry.
Oh.
I was like, oh.
So it's just like.
There you go. Is it like that? Do you have your pipette at 10? Yes. Okay.
Because we had a quiz earlier.
Nah.
Everybody was able to take a quiz. So everything is set up for you. You can just unwrap it and the instructions are over there. So whenever you're ready.
Yeah.
We don't need it anymore, but.
Beans.
Oh, you know.
No, do you guys still need it?
Not right now. Okay, so
You know what?
Give me that half
Here's an
of our wells
idea.
are broken.
Luis is going to set up an e-gel. If yours really does not work for some reason, you can reload them.
Whoa, my hand's so shaky.
That was like 1 year.
No,
Push
yes.
down together with
Just run it once. Just run it once here. Okay. By the way, you have a pepper right now. Because it seems to be, it was partially filled.
It was from the bubbles.
Yeah.
We tried to get all of them out, but it.
Oh no, that's one the one I stabbed into the last one to see just how deep the well was.
Yeah, good you.
But I was wondering if I just wasn't getting it stabbed in, but.
You good?
The loading diamond here, man. Get some more loading.
Alec, before you continue. There's a little tip.
That was another thing.
Nothing yet.
Use one of your fingers as kind of like a guide. Because you have them basically like this, and before you know it, it's static fluid. Like use your finger as a guide and do it not like completely like this, but a little bit tilted. And with your finger, put your finger a little lower, close to the tip. Exactly like that. Yeah. That way you have way more control over it.
Great. I'm like sliding out of my seat.
You want to move that one up?
And easier?
There you go.
Yeah, it's the wrong direction.
As long as you can see the band and it doesn't dissipate, stays in there, you're fine.
It might look a little weird.